Hello, everybody. A very good afternoon. I once again welcome all our participants, distinguished viewers, and dear students in the second day of our webinar entitled Walk Through the COVID-19 Pandemic, Viewing by the Lenses of Researchers and Commoners. In yesterday's session, although we had some technical problems, but in spite of that, I am happy to declare that we received an overwhelming response from a large number of participants from our state as well as from other states of India. We had 534 registered candidates and over 1200 views in yesterday's session. Today also we hope to receive the same. In today's session, we are happy to have with us two very well-known young faculty and researchers as speakers, Dr. Anirban Bhor and Dr. Gaurav Sarkar, who shall share with us their scientific understandings and boost our moral spirits for winning the war against COVID-19. Now, to introduce today's first speaker, I am extremely happy and contented to introduce Dr. Anirban Bhor, who received his master's degree in botany with specialization in microbiology and biotechnology from University of Bardhaman in 2008. He then joined Bose Institute Division of Plant Biology with the prestigious Junior Research Fellowship Award from CSIR under the supervision of Professor Shampa Das and obtained his doctoral degree in 2017. But prior to receiving his doctoral degree, he joined Ramakrishna Mission Vivekananda Centenary College, Lahore as the assistant professor in the Department of Botany in 2015. Presently, apart from being a faculty in the Department of Botany, he is also serving as the joint coordinator of Department of Microbiology, RKMVC College, Lahore, Kolkata. Besides, he also received a prestigious postdoctoral fellowship funded by the European Union and worked as a postdoctoral visiting scientist at Natural Resource Institute, Finland, and Wiki Plant Science Center, University of Helsinki, Finland, from 2018 to 2020. He is also the recipient of University Gold Medal and Jawaharlal Nehru National Merit Award, Government of India 2008. His doctoral work focused on unraveling the molecular intricacies of plant microbe interaction between chickpea and Fusarium oxysporum, FSP size series race 1, using both molecular techniques and bioinformatic tools, while his postdoctoral work was involved in deciphering the hormonal signaling networks operational during lignin biosynthesis in the first sequenced and completely annotated gymnosperm, Picea avis. His present research involves characterization of different endophytic microbes having potential roles in human welfare. Today, Dr. Anirban in his talk titled What We Have Learned from COVID-19 Pandemic, Pragmatism Over Panic, shall help us understand the behavioral features of COVID-19 within the human host and the possible measures which we can adopt to boost our inherent immunity and put an early stop to the viral entry. So, over to Dr. Bhor.
Thank you. And good afternoon. At first, I want to express my gratitude to all the organizing committee of RMV for organizing this national webinar on walk through the COVID-19 pandemic, viewing by the lenses of researchers and commoners, and giving me the opportunities to interact with all of you. And today, the topic of my talk is what we have learned from COVID-19 pandemic, pragmatism over panic. The COVID-19, as the name implies, is a coronaviral disease and is, as we all know, that
Hello.
Transmitting so quickly into the population. So before I
I'm sorry, I guess we are having some more technical issues. I request all the participants to please have their questions emailed with us.
think the genetic material, either DNA or RNA, are protected by the surrounding protein in core, which is called the capsid. And this whole simple structure is again covered by another lipidus envelope. And this entire structure is has a specific symmetry, either helical, isosceles, or complex symmetry. And these viruses can infect, ranging from the bacteria to the higher organisms like human beings. And the size of the viruses varies also very widely. As you can see, the variola has only the 20 nanometer in diameter. And the coronaviruses have the diameter of 120 nanometer in size. And in the next slide, you can just see how this virus, the small viruses, can be compared with the larger size bacteria and the human red blood cell. So you can see that it's a very tiny structure, and it can, as compared to the bacteria and the human red blood cells. So the question arises that how these teeny life forms can control over this most advanced life forms like human beings, the multicellular organisms. The answer is the size does not matter. What matters is the number. We all know that when the life forms are very much simple and teeny, they have the quite higher capability of reproduction. They can multiply quite high in number. So within the cell inside, like a human cell or any other host cell, these viral particles can multiply rapidly to produce a huge number of uh, infecting particles, and that actually control over the host cell. And in case of the coronavirus, the around 1,000 viral particles is enough to get infected a healthy individual. And once it comes in contact with the body, it takes only 10 minutes to get inside the body. And I will come in detail regarding these two facts later on. And now, a brief family background of the virus. This coronavirus belongs to the order Nidovirales, and under the Nidovirales, there are four different types of coronavirus are categorized, and they are alpha, beta, delta, and gamma coronaviruses. The delta and gamma coronaviruses are mainly in avian origin, whereas the alpha and beta coronaviruses
Hello. I guess we are having some technical issues. I request all the participants to please have their patience. Technical support is trying to resolve the problem. I request the participants to please bear. Okay, I'm sending you his number. You can please contact.
Hello, am I audible? Hello?
No, your connection went off in the middle. Could you please go back to your slides? Your connection went off in the middle. Could you please go back to your slides and show once again?
Hello? Hello?
Hello? Can you hear me, Dr. Bhar? Can you hear me? Hello? Dr. Bhar, can you hear me?
Hello?
Hello? I guess you have lost the connection with him. Hello?
Hello? Hello, am I audible?
Hello?
I can't hear anything. Hello?
Hello? Can you hear my voice, Dr. Bhar?
Am I audible? I can't hear anything.
Hello?
Hello?
Yes, we are being able to see your slides, but your voice is not clear.
Hello? Hello, I can't hear anything. Hello, am I audible?
Hello? Hello? Yes, your slides are being viewed, Dr. Bhar, but your audio is not clear.
it clear?
You
Is it
please
clear now?
mute. You please mute your telephonic connection.
Hello?
Hello.
Hello now?
Hello. Can you hear me, Dr. Thor?
Is it clear now? Hello?
No, I'm sorry to say your voice is still cracking. I'm sorry to say
I can
that
hear
your
you, but
voice is
I
still
don't
cracking.
understand why people can't hear. Because everything is clear here.
Yes, Prabhuji.
Hello? Hello? Is it clear now? Am I audible? Hello?
Hello. Your slides are clearly visible from the audience, but you are not audible.
Am I
You remove your
audible?
headphone
Am I
and
audible now?
please remove your headphone and try to carry on with your
Hello?
slides. Sir, please remove your headphone and try to carry on your slides. Your slides are being visible.
Can you repeat?
Hello? Hello?
Okay. So
You speak,
now how it looks?
sir. Yes. You
Level
go
of virus
on
is
speaking. You speak in
so clear what we
a
can say, and
continuous low voice. It will be
the
audible.
answer is ranging from 60 to 140 nanometers. And the positive standard RNA virus is with unusually large RNA. And under the microscope, it looks like a ground in presence due to presence of the glycol. Under electron microscope, it looks like a ground due to presence of glycoprotein in the envelope that produces a cloud-like appearance. And the diameter of the SARS-CoV-2 is 125 nanometer and the spherical insect. The symmetry is helical, and unlike other coronavirus, which is sensitive to UV light and high temperature, like greater than 60 to Celsius temperature, and it can also survive in chill up to 0 degrees Celsius. The liquid solvents can effectively degrade the virus, and the chlorine containing solution, sodium hypochlorite, peroxyacetic acid, and chloroform, etc., can readily kill the virus. And it has been shown that the chlorhexamide can also limit the viral load. Now, is a general schematic uh, representation of how the virus enters the host cell and take control over them. First, the viral particle comes in contact with the receptor and it interacts with the receptor
And then, after successful recombination, reaction the viral genomic material enters inside the cell, and it utilizes the transcription machinery of the host to produces replication enzymes, and that produces large number of the genomic material and throws the back into real viruses. And in doing that, the host immune response and constant selection pressure and also additional stimuli like drugs and other things can causes the mutation and it causes the other forms of the virus which may be more virulent or less virulent. But after the production of the new viral form, the cell of the host cell are disintegrated and a necrotic pool is generated. And that is the origin of the secondary infection. The coronavirus uses similar human receptors, and our SARS-CoV-2 is utilizes the ACE2. The SARS-CoV-2 utilizes the ACE2 or angiotensin converting enzyme 2 as a receptor, and it is the structure of the coronavirus. This is the structure of the coronavirus where we can find the spike proteins, nucleocapsid, membrane, and envelope. And the most important part of this four structural proteins is the spike set or capsid spike protein. And this spike protein interacts with the ACE2 enzyme and produces a receptor RBD ACE2 interaction. The genome organization. The genomic organization tells that the spike protein, membrane protein envelope, and nucleocapsid are the major structural portion. And if we look into the structure of the genome, the replication enzyme or the enzyme which are utilizes the translation machinery of the host and produces the proteins for the uh, produces the proteins for the viral organization. And the coronavirus structural proteins are relatively large in size, and there are several modifications is possible in this region, and that's why the generation of vaccine is quite difficult due to the huge probability of modification in the structural region. The SARS-CoV structural organization, you can say the nucleoprotein has 1,000 copies of the envelope proteins, which produces the envelope of the 20 copies, and the membrane proteins have the 2,000 copies. What is the spike protein?
Hello, sir. No, 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 no. He is also facing the same amount of problem. Network problem. I think you talk to the technical person and then decide because I am already live. Okay, okay, sir. On behalf of the entire organizing committee, I apologize because we are having serious technical issues, and as you all believe that this kind of webinars, we are solely dependent on the technical support, and internet connectivity is such a thing we really don't have hands on. So our technical staff is solely trying to resolve the issues. However, we request all our participants to kindly have patience. We all believe we are aware of the fact that post Amphan, the situation has not completely resolved, and the central part of Kolkata is still facing serious problems regarding network connectivity. Yes, I can understand the participants are becoming impatient and they're asking us to solve the problem, but I sincerely apologize and I hope you will understand this is a problem which you have to solely believe on a connection which is unmanned. It is really not our part and not the speaker's fault also. I request all the participants and I declare with due permission with our authority and IQAC coordinator, as per the consent from our principal sir, Professor Bhattacharya, Professor, hello, Dr. Prashanta Bhattacharya, our principal sir, and IQAC coordinator, Professor Tanmoy Bandyopadhyay, with their consent, I think we have to reschedule the program. Hello.
Hello,
We have,
yes, you are audible. You are quite audible.
hello.
You are audible.
Sir, will we go with the second speaker? Will we go with the second speaker, sir?
Yes.
Okay, okay, try this. If it really works out.
Okay, with their due permission, I think we will start with the second speaker. We are having serious issues with. Okay. We are giving it a try. Now I request our second our speaker, Dr. Gaurav Sarkar. I request my joint coordinator, Professor Joita Shaha, to give the introduction of Joita Shaha to introduce Professor Sarkar.
Good afternoon, everyone. Am I audible? Ronobi ma'am, am I audible?
Yes, you are audible.
Ma'am?
Yes, you are
Am I
audible.
audible?
Yes.
Okay, thank you. Okay, thank you. Good afternoon, everyone. Now, I warmly welcome our next speaker, Dr. Gaurav Sarkar, who is working as Deputy Wellcome Trust India Alliance Fellow in the Department of Botany, Shikha Bhavan, Vishwabharati. Dr. Sarkar obtained his PhD in immunochemistry and molecular allergology in the year 2017 from Bose Institute under the supervision of Professor Swati Gupta Bhattacharya. His research interest includes molecular and mediated. Ronobi ma'am, am I audible? Am I audible? Ma'am, am I audible? Am I audible?
Yes, you are audible.
Okay. Okay. Sorry for the interruption. So, starting with the proteomic identification of diagnostic important respiratory and food allergens, he continued to develop recombinant allergen-based diagnostic as well as therapeutic allergy vaccines. His strategy of developing recombinant allergy vaccines is based on epitope mapping, followed by the design of genetically engineered hypoallergens. His future goal is to advance these hypoallergenic vaccines into clinical applications. In his current project of India Alliance Early Career Fellowship, he is working on serological detection of major gonidial antigens from Aspergillus fumigatus. His present lecture encompasses the ongoing efforts being made to develop structure-based recombinant vaccines against COVID-19 with an aim to contain and limit the spread of the disease. We wish his lecture will enlighten the path of journey from structural biology to vaccinology of novel coronavirus. Additionally, we expect the present talk will arouse positive hopes in human minds toward achieving effective drug targets against this infectious disease. So, over to Dr. Sarkar. Over to sir.
So am I audible? Can you hear me?
Yes, 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 sir. Yes, sir,
Okay,
you are
so should
audible.
I start? Okay,
Yes,
so
sir.
should I
Yes, sir.
start?
Yes, sir.
Okay, so I
You
will
can start.
be okay. So I will be explaining each slide, and Dr. Porel from the technical part, she will be changing the slides whenever I will ask her to change. Okay.
Okay, okay.
Okay. So thanks for the introduction, nice introduction, and so I'm going directly into my topic. The title of my topic is "Combating the Global Pandemic COVID-19," and as a journey starting from structural biology, what we learned from structural biology, and then going into developing a better vaccine to combat the disease. So I think yesterday our previous speaker has already delivered a great deal of information about the structure of the SARS-CoV-2, the virus, the anatomy of the virus. So I'm just giving a brief introduction about the structure. So it's a spherical one. It has protruding the spike proteins on the surface. That's why it's called corona. It looks like a crown, and then it has a waxy layer which is made up of lipids from which some hydrophobic proteins are protruding out, are projecting out on the surface, and there is a core where the genetic material of the virus is packed. And this genetic material is basically a single stranded RNA which is in complex with the RNA, the nucleocapsid protein, and that's how the virus looks like. And the diameter of the virus particle is about 125 nanometer or to 130 nanometer. And this virus, the virus, the next slide. Please go to the next slide. Yeah, the third slide, slide number 3. Yeah, so the virus causes the disease called COVID-19, and the disease is basically a respiratory distress associated with different flu-like symptoms. And the first line treatment for COVID-19 for this disease is totally supportive and symptomatic. That means there is right now there is no medication available to kill the virus, and the supportive treatment includes the supplemental oxygen therapy, giving respiratory support to the patient, fluid replacement, body fluid replacement, and then use of analgesics and antipyretics, and managing secondary infection by administering antibiotics caused by bacteria. So these are the supportive treatment that can be helpful to manage the disease. And in addition to this supportive treatment, we can also there are a number of the names of a number of certain drugs are coming up, which people have started thinking that they could be used as wonder drug. They can cure the disease, but to be honest, these drugs are not specific for this virus. They don't specifically kill the virus. Actually, these drugs were originally developed for some other. They are meant for some other pathogens, like HCQS was is meant for the malaria parasite, Plasmodium falciparum, and these three, flavipiravir, remdesivir, and rotenavir. These three are basically antiviral agents, and they were
the flavipiravir was developed for treating influenza virus, the remdesivir for Ebola, and Marburg and Ritonavir for HIV protease to inhibit HIV protease. But they don't specifically, this flavipiravir and remdesivir, these are they inhibit the ribonucleotide polymerization, and they don't actually kill the virus, but they inhibit the viral propagation by inhibiting the RNA polymerase, and thereby giving the host immune system enough time to clean up the remaining virus particle from the system. And there are other two drugs which are available. This is called the anti-interleukin 6 antibody. This is a basically biological drug. It's very expensive, and this is actually developed to manage the symptoms of autoimmune disorder like SLE, rheumatoid arthritis, and it is believed that this can be helpful to manage the cytokine storm that comes after the viral infection. So this is also not specific for this SARS-CoV-2. And very recently, the name of another drug is coming in the news. It's the dexamethasone. This is basically a corticosteroid, nothing else, like deflazacort and other corticosteroids, which helps to suppress the immune system. That is immunosuppressive drug, and thereby it also, just like anti-IL-6, it also helps to reduce the severity of the symptom. Okay, the next slide. So, in addition to other than this symptomatic or first the supportive treatment, right now the only targeted approach or the disease modifying approach is by developing a antiviral drug specific for the coronaviral group, the SARS-CoV-2, or by developing a specific vaccine. So for developing a drug, we need a detailed knowledge on the viral physiology, the strategies of viral replication, invasion into the host cells, infection, and how they release from the cells, the entire viral physiology. But and it takes a whole lot of time to understand how the virus works within the host system. And in case of vaccine, we need certain amount of the knowledge of immune response or the host immune response after infection. And right now we have some certain degree of information about the immune response after COVID-19 infection, like the cytokine storm, how seroconversion occurs, how frequently, and what are the interferons and the other cytokines that are modulated, upregulated, or downregulated during the infection. So we have certain degree of knowledge, especially from the previous SARS-CoV-1 infection. And so it's a good it's good to think for a vaccine, making a vaccine for this COVID-19, because it might take less time than developing an effective drug specific for the coronavirus. So what is vaccine? Vaccine is basically either an intact pathogen, the pathogen itself, or a small piece of the pathogen, a small molecular piece of the pathogen that can that can boost immune response in the host by boosting or by triggering the humoral response, that is antibody mediated response, or the cellular response. And these are the key features of an ideal vaccine. The broadly neutralizing antibody, that means because as the this SARS-CoV-2, this virus is highly mutagenic. There are multiple strains available across the globe. So an ideal vaccine should induce broadly neutralizing polyclonal antibody that can neutralize, that can clean uh, a broad range of strains, viral strains under this SARS-CoV-2. The second point is safety. It should be safe for the patients, for the human system. There should not be any adverse effect. Then the durability, that is the duration of the immune response, how long the immune response lasts. For example, in case of the tetanus vaccine, or in case of the flu shot, influenza vaccine, we need shots in every year, because the immune response, there is a robust immune response, but it is not long lasting. The immune response goes up and then declines over time. And again, there are certain type of vaccines like polio vaccines, which is or hepatitis vaccine, hepatitis B vaccines, which are administered only once in a lifetime. And the memory cells are quite good enough to remember, to memorize the pathogens. So the immune response lasts forever throughout the lifetime. Then the number of doses, the dose should be as less as possible. So an ideal vaccine should have as minimum dose as possible. And the fifth is the manufacturing cost, that is the purchasability, because the vaccine should be such a vaccine that can be available to the poor country, the third world countries. So the manufacturing cost is another point, another thing that should be taken into consideration. Okay, the next slide. Now, this is how the vaccine works. This is generalized overview how the vaccine works in our system, in human system. So the first part is the antigen presentation. So when the vaccine is administered, there are certain group of cells, immune cells, which are called the macrophage or antigen presenting cells. They take up the vaccine and then they chop it out into small pieces by proteolytic degradation. And these pieces, the peptide fragments, are called the T cell epitopes. These T cell epitopes are then presented on the surface of the antigen presenting cells along with a specific type of receptors, which are called the MHC, the major histocompatibility complex molecule. These are the surface molecules that are presented on the antigen presenting cells. And they are also known as HLA, human leukocyte antigens. And there are three types of HLAs, HLA 1, 2, and 3. 3 doesn't play to that much significant role in viral immunology. The one, HLA1, that is expressed in all type of cells, and HLA2 are expressed in only in the professional antigen presenting cells. And this HLA, there are high degree of genetic polymorphism in HLA1, that is the MHC1 molecule. And this is the reason why a certain group of people are highly predisposed or highly susceptible to severe infection. And that could be an explanation of why the United States have such a high degree of fatality rate than India. So this is the first step of the vaccination or immunization with vaccine. In the second step, this is called the T cell activation. So what happens in T cell activation? These macrophages or APCs with T cell epitopes presented on their surface, they interact with the T cells or T lymphocytes, this type of white blood cells, and they have T cell receptors on their surface. And this interaction results in uh, differentiation of different lineages like cytotoxic T cells, the suppressor T cells, and the helper T cells. This helper T cell is very important because that ultimately trigger the antibody response, the protective antibody response. So what happens? These are also called TH cells. The helper T cells are called the TH cells. So what happens in the next step? That is the B cell activation, in which the antibody is produced. So the B cell, in the same way, the macrophage engulfs the vaccine, the same way the B cell endocytoses the vaccine particle, and then they, in an MHC independent manner, that is unlike macrophage. The macrophage, they engulf in MHC dependent manner, and the B cell engulfs in an MHC independent manner. And the vaccine is then again degraded into small pieces called B cell epitopes. These are small peptides, which basically the small linear epitopes. And these epitopes are then presented with MHC. So the engulfment is an MHC independent manner, but they are presented along with HLA, these B cell epitopes. And these B cells with the B cell epitopes presented on their surface, they interact with the activated T cells, and that results in the release of lymphokines, a certain group of chemicals or chemoattractants from uh, from the T cell, and these lymphokines stimulate B cells to differentiate into either plasma cells or the memory B cells. So memory B cells, they are the basic of the immunological memory. They memorize the pathogen so that they can boost immune response during a second encounter. And thus another lineage is called the plasma cells. These plasma cells are basically they are derived from B cells, and they are antibody producing cells where the HBJ recombination occurs, and specific antibodies means the antibodies specific for the pathogen are produced. And these antibodies then they are secreted into the circulation, and they help they keep our body safe from the natural pathogen. Okay, the next slide. Now, these are the steps of vaccine development. First step is the exploratory stage where the basic research takes place. That is to select the vaccine candidate, the molecule, uh, which should be used for immunization. This is what the basic scientists are doing. Next come the preclinical stage. The preclinical stage is the stage where the animal model testing is done, or the testing on the cell lines are done. And once the preclinical stage is qualified, then the vaccine enters into clinical stage or the clinical study. And in the clinical study, there are three phases: phase 1, 2, and 3. In the phase 1, there are a certain group of human beings or participants are included. In phase 2, uh, a large number of participants or volunteers are included, like hundreds to thousands. And in phase 2, it's a huge population of the participants in hospitals. The tertiary care hospitals are included to test the vaccine. And the purpose of the clinical clinical trial, is to assess the safety, the the safety, and then to standardize the dose, to standardize the formulation of the vaccine, and to, of course, to study the efficacy, that is how efficiently it can trigger immunogenic response in each of these three phases. And once all these trials, preclinical and the clinical trials, are qualified, then the vaccine gets necessary approval from the competent authority and the government. And here I have stated how many times it should take for each step, in each step. So like preclinical stage, an ideal preclinical stage can take 5 to 10 years. Clinical development can take 3 to 8 years. However, in the recent years, for example, in case of COVID-19, uh, there are an accelerated kind of clinical trial is going on, or preclinical or clinical trial is going on, as we are there is an urgent need of vaccine. The next slide. So there are different types of vaccines: first generation, second generation, and third generation. The first generation are the very old type of vaccines which were used, like this mumps, MMR, mumps, measles, and rubella, and then the polio vaccines. So first generation vaccine, the whole agent, the whole organism, the whole pathogen is injected or ingested through mucosal route. And in that case, the pathogen is either killed, inactivated, heat killed, or inactivated by using formalin, or it can be live attenuated. Attenuated means, so by doing some genetic manipulation of the pathogen, the virulence of the pathogen is decreased or attenuated. And there are some loopholes for first generation vaccine, because especially for the live attenuated vaccine, because they can sometimes, this live, as they are live, the pathogen is still alive, so they can revert to its original form inside the human body. Once they are inside within the human body, so they can revert to its original form. So there are risk factors for the first generation vaccine, especially for the live attenuated vaccine. And the BCG vaccine, which is used to used for tuberculosis, MTB. That is an example of live attenuated vaccine. Then comes the second generation vaccine. Here comes the application of the recombinant DNA technology and the protein structural study of the proteins. So in that case, what happens? Not the intact pathogen, but a molecule of the pathogen, a protein or a glycoprotein, surface protein especially, is used as the vaccine, and it is, of course, it is safer than first generation vaccine, because there is no chance of the pathogen induced disease onset, because it's just a molecule. And they are usually either the protein, intact protein, or they can be engineered by recombinant DNA technology by removing certain parts of the protein, just a small part of the antigen, recombinant antigen. And also there are some peptide based vaccine, just small, a 20 or 40 amino acid residue long fragments of the antigen itself. So these are the peptide based vaccines, and these peptides, sometimes the B cell epitopes or the T cell epitopes can be included in the peptide based vaccines. And then comes the third generation vaccines. These are, I have written DNA vaccine, but it's better to say nucleic acid based vaccines, where the DNA or the RNA, that is the genetic material itself, is used as the vaccine, and I'm going into details of that in my following slide. So for the second generation vaccine and the third generation vaccine, we need detailed knowledge about the molecular structure of the protein or of the antigen, which will be used and immunogen in the vaccination schedule. So here comes the application of structural biology. Next slide. So so right now these are the three proteins for which three proteins of the SARS-CoV-2 virus, the novel coronavirus, which have been the structure have been solved at atomic with sufficient atomic details around 2 angstrom, with a resolution of 1.5 to 2 angstrom. These three proteins are spike proteins, the papain-like protease, and the RNA-dependent RNA polymerase. Out of these three, the spike protein is the only structural protein, and the other two are the functional proteins. And the protease, it helps in viral assembly, and the RNA polymerase, it helps to replicate the genomic material. And the spike protein is a structural protein which helps a virus to gain access into the host cell. And out of these three, the spike protein has been selected as an ideal candidate to make the vaccine, because it is the most surface accessible protein of the virus. This is an ideal vaccine should have enough of the solvent accessibility, should be on the surface. And it is also, there are some sugar moieties associated with the spike protein. So all of this make the spike protein an ideal candidate for vaccine development. So this is how the spike protein looks like. This is the primary structure. The next slide, sorry, the next slide. This is how the spike protein looks like. This is the primary structure of the spike protein. So there are two domains, S1 domain and the S2 domain. The S1 domain is approximately 200 kilodalton in size, and it has another domain is called the RBD domain, or the receptor binding domain. And the S2 domain is the next part, where the fusion, which helps in the fusion, that is fusion of the virus particle with the host cell, the virus membrane with the host membrane. So this is the FP, is the fusion part, is the domain which is responsible for the fusion. And there are HR1, HR2, these are heptad repeats, heptad 7, I think it's 7 amino acids long residues. And there is these arrows indicate the cleavage site. So after binding with the receptor, I think my previous speaker has already said how it works. Protease, there are proteases which first cleave the spike protein, and then the spike protein becomes active, the fusion domain is exposed, and then it binds with the ACE2 receptor and helps the membrane fusion. the fusion of the virus, the membrane of the virus and the membrane of the host cell. So this is the primary structure of the protein, and this is a schematic diagram below. This is a schematic diagram of the spike protein, how it looks like. So this is the transmembrane domain in the bottom, the pin-like structure. This is the the transmembrane domain. It passes through the membrane, and it's made up of hydrophobic protein. And then this is FP is a fusion domain which helps this the orange, the red one is the fusion domain which helps in fusion between the the membrane of the virus particle and the membrane of the host cell. And the magenta color is the RBD, that is the receptor binding domain, which helps the viral particle or the virus particle to gets attached on the S2 receptor. So this is the overall structure of the spike protein. And this RBD, the receptor binding domain, is highly flexible. In case of SARS, in case of spike protein, this the receptor binding domain is highly flexible in case of the novel coronavirus and the SARS-CoV-1 in both the virus, as compared to the other members of the coronavirus group, like the alpha coronavirus, beta coronavirus, and the gamma coronavirus. And this flexibility can be associated, can be thought, we can think that this flexibility is the reason behind that much of infectability or the virulence of these two strains of coronavirus, the SARS-CoV-1 and the SARS-CoV-2. And also in case of SARS-CoV-2, certain mutations have been identified in the spike protein which are different from the SARS-CoV-1, and these mutations, amino acid residues like 5 or 6 residues, which help the SARS-CoV-2, which are thought to help SARS-CoV-2, this novel coronavirus, to get firmly attached with the receptor. And that is another reason why this novel coronavirus is so infectious than the previous SARS, the SARS-CoV-1. The next slide. So how the spike proteins is being used as a vaccine candidates by different approach. So the spike protein, intact spike protein can be used, the full length spike protein, as a vaccine, and it has got some disadvantage because it can impair the liver function. However, it is very effective for inducing neutralizing antibody because it is a intact S protein, the whole S protein, and it can also trigger the cellular immune response, the T cell response. And also there are recombinant S protein, the third one, the recombinant S protein based vaccine. That means not the entire S protein, but by using genetic manipulation or genetic engineering, certain domains of the S proteins are removed, and they are associated with the remaining part is then attached or fused with some immunogenic agents or immunogenic proteins, like for example the pre-S protein of the hepatitis B virus, or it can be KLH, the keyhole lymphate, that immunogenic KLH immunogen. So this is another type of this can be another approach for making vaccine with the spike protein, or it can be just solely the RBD, that is the receptor binding domain of the spike protein. And also there are this second and the third one, these are the nucleic acid based vaccines. So this can be the DNA based vaccine or the mRNA based vaccines, and I'm coming into that. So each of these approach has got its own advantage and disadvantages. So it's all about trial and to wait for the results, how each of these approach works for in case of the COVID-19. And in the below I have given the link in which you can find the currently ongoing projects of the COVID-19 vaccine development, different approaches all around the globe, and their current status, in which phase trial they are. Okay, so now, so next slide. So in this slide, I will discuss how the viral vector-based vaccines work. So what happens? So I think you have heard about the Shadox vaccine that is being developed by the University of Oxford. This Shadox vaccine, this is actually this the it stands for the chimpanzee adenoviral vaccine developed by Oxford University. That's from and therefore the name that's what the name stands for, the Shadox vaccine. So the adenovirus, this is basically a viral pathogen for chimpanzee, and this adenovirus is with the help of genetic manipulation, the adenovirus is then weakened or modified to reduce the virulence properties, the virulence factors of this virus, and then this virus can be used as a vector, that means the carrier of the genetic material that encodes a vaccine. So what people are doing in Oxford University, they are taking the spike protein, the genes of the spike protein, and they pack the virus, this modified adenovirus particle, with the genetic material which codes for the spike protein, and this is how the vaccine is developed. And this carrier, I mean this entire particle, the virus particle, less virulent virus particle packed with the genetic material encoding the spike protein, is then injected in the human body. And once inside the human body, in the lower diagram, once inside the human body, this you can see how the genetic material is designed. It is a coding sequence, the open reading frame that codes for the protein, which starts with a promoter that helps in transcription, that there is a gene expression, and there is a stop codon, of course. And once inside the human cell, like muscle cell or where it is injected, this genetic the genes or the ORF coding starts coding for the protein. I mean, the translation starts inside the host cell, and so this is you say in situ synthesis of the protein inside the host cell, where the protein is uh, not directly injected, but the genetic material coding for the protein is directly injected, and the protein is being synthesized within the host cell. And once the protein is synthesized, then they are presented on the surface, and the usual pathway, how I mean the usual pathway of vaccine induced immune response takes place. Okay, the next slide. So in the next slide, this is the example of another type of our nucleic acid based vaccine that is being developed by the Moderna, by a company, a pharmaceutical company called Moderna. It's a US based company, and in association with the National Institute of Health. And this is another approach of making nucleic acid based vaccine, in which the viral vector is not used, and instead of viral vector, the lipid nanoparticles are used. That's called the LNPs or liposome. So the genetic material is directly packed within the LNPs, and this entire structure, the LNPs packed with the genetic material, is then injected as a vaccine. And once inside the cell, the genetic material comes out of the LNPs, and they start making proteins by usual translation, by using the translation machinery of the host cell, and the proteins or the antigens are thus produced. And again, the immune response, the immune pathway starts once antigen is produced in its usual way. So this is how the viral vector-free vaccines are being designed. So the basic difference between the adenoviral vaccine and the LNP-based vaccine is the basic principle is the same, in which the genetic material encoding the antigen or the immunogen, that is a vaccine, is being used. This is the active ingredient for for both the cases. However, the vector is different. In case of adenoviral based vaccine, adenovirus vector based vaccine, a virus particle, attenuated or weakened virus particle, is used as a vector. And in this case, this mRNA 1273, this is under phase 2 trial, I think, for COVID-19. So in that case, the LNPs, lipid nanoparticles, are being used as a carrier, as a vector for delivering the vaccine. So it's all about, so I mean, I have mentioned these two types of vaccines, the nucleic acid based vaccines, as these are the third generation vaccines, and people are interested to know about the how it works. So so there are many, still there are different types of vaccines, the protein based vaccines, also the heat killed attenuated vaccines are being developed by various researchers across the globe for COVID-19, and all the informations are available in the link which I have provided, the update about the vaccine trials. Okay, the next slide. So these are the loopholes or the shortcomings that can happen during the vaccine development process. So people are expecting that vaccines are can be available by the beginning of the next year 2021 or the mid of next year, but still we should not be over optimist about the availability of vaccine, because a vaccine, there can be lot of problems can arise during the vaccine development process. For example, the number one, no immunogenicity can happen after preclinical trial. So preclinical trial, that is using the mouse model, animal model, or the non-human primate model, a vaccine can be found safe and immunogenic in animal model, but in after animal model, after the preclinical trial, it might not show any immunogenicity or efficacy in human. So there is a very common saying in scientific community that monkeys lie and mice do not tell the truth. So we should not trust or we should not rely too much on the preclinical trial data. The second shortcoming is about that safety and the toxicity. So a vaccine can be fine, can work fine with animal model, but it can show certain degree of toxicity in human. For example, the S protein is thought to impair liver function. It can be toxic for the liver function. So there are safety issues. So a vaccine which can induce protective immunity could be highly toxic for human body. So that is another shortcoming. Third is the less durability, that is the durability of the immune response of the vaccine. So there are vaccines, there can be a vaccine can be totally safe for human being, and it can show high efficacy, robust immune response against a pathogen. However, the duration of the immune response, that is another important point, because we cannot immunize a person 3 to 4 times in a year. So the duration of the immune response, that should be taken into consideration. That is another point, another shortcoming that can happen during vaccine development. And finally, that is very important point, that is called ADE, or antibody dependent enhancement of the infection. So a vaccine candidate can induce immune response, can induce antibody response, humoral immunity. However, that antibody, instead of giving protection, can enhance the severity of the infection. That is called the antibody dependent enhancement, or sometimes it is called immune enhancement. That was found once it was found in the vaccine trial for the dengue virus. So that is another very important issue that should be taken into consideration. So these are the factors, key factors, that can result, or that can lead to delay in the availability of an ideal vaccine. Okay, next slide. So these are the solutions what people, the government, and the healthcare professionals have been saying from the very beginning. So these are the solutions before the availability of the vaccines. We should be very careful about our personal hygiene, using mask and sanitizing our hands. Then physical distancing. I would prefer to use the term physical distancing rather than using social distancing. So physical distancing should be maintained, but the social cohesiveness should be intact. And finally, strengthening immunity. There are many tips available which can help to strengthen our immunity, like having vitamin C, having fresh vegetables, having enough adequate water in a day. These are the helpful to having enough rest. These can be helpful to strengthen our immunity. So that's all about my talk. So the next slide. So that's all about my talk. Next slide. Okay. Okay, I'm done.
Yes, thank you very much, Dr. Gaurav Sharkar, for your great delivery and also helping us in understanding the power and also the limitations of drug and vaccine design and delivery. So, as we all know, that now we are having a question answer session, but due to technical problems, we believe we have made a redesign of our program, and we will shift the entire question answer session at the end of the session. As we all know that due to technical problems, Dr. Dhor's talk we did not hear, but now he's back with us in the studio, and initially we will hear his talk also, and then at the end of the session, we will have the question answer session. So, as I have already introduced Dr. Dhor, so without wasting any time, I ask Dr. Dhor to present his talk. Please, Dr. Dhor.
Thank you. And I'm extremely sorry that for the inconveniences happened earlier due to some technical fault. And I don't know how far I, uh, I was audible in the earlier, so it's better to start from the beginning. Uh, so I want to thank all the organizing committee for inviting me, and it's my great pleasure to interact of all with you. And the topic of today's seminar is the COVID-19 pandemic viewing by the lenses of the researcher and commoners is a very contemporary event, and we all know that the humankind is facing a difficult challenge, the most difficult challenge since last 6 months. And the topic of my today's talk is what we have learned from COVID-19 pandemic, pragmatism over panic. Next, next slide. So first, as uh, as all the introduction has given by my previous speaker, and the viruses are the ancient life form, that the simplest, and at the same time the simplest life form that have the genetic material, either DNA or RNA, covered by the nucleocapsid, and it's again covered by an envelope which is lipidous in nature, and that's why the soap and the alcohol-based hand sanitizers can kill the virus particle because of this outside lipidous envelope. It destroys readily with come in contact with the either soap or alcohol-based hand sanitizers. And now in this slide, I want to show you that how the different sizes, the variation of the different sizes of virus are there. The largest one, that is the thread-like structure, is the very well-known virus, is the Ebola, is a it's a very large in size, and the lowest is the smallest size is the 20 nanometer parvovirus. So there is a huge variation in shape, and the size of or diameter of the coronavirus is ranging at around 120 nanometer. Next slide. And this slide shows you the comparative size of the smallest teeny life forms, that is the virus, with the bacterium like E. coli and also the human red blood cells. So this is a very teeny life form that take control over the mostly evolved human beings and the multicellular other multicellular organism. Next slide. Next slide, please. So the question is how these teeny life forms can control over the huge multicellular organism. The answer is that the size doesn't matter. What matters is the number. And we all know that when as the life forms become simple and the low and the like primitive organism have the greater capability of reproducing. So this viral particles when enters inside the cell, it can multiply in a rapid basis to produce a huge viral load, and that is enough to control the entire human body or the multicellular organism. And for the COVID virus, at around 1,000 viral particles are enough to uh, the 1,000 viral particles are enough to create an infection. And once the viral particle comes in contact with the human body, it takes only 10 minutes to get entry inside the host body. Next slide. So this is a very brief family background of the coronavirus. You can see it belongs to the family Nidovirales, and there are four different categories of the coronavirus so far reported is alpha, beta, delta, and gamma coronaviruses. And the right side, the delta and gamma coronaviruses are the avian lineage, and the alpha and beta coronavirus are of bats and rodents origin. Next slide. Next slide, please. So, till date, 7 human coronavirus or HCOV have been detected, and the first human COV was detected in 1960, and all the other 6 are identified in this millennium. And it is very interesting to note that the human coronaviruses that have the origin from the alpha coronaviridae group, that is the HCOV OC443, HCOV 229, and HCOV NL63, produces a very mild symptoms like common cold like symptoms, and they because they have the self-limiting upper respiratory tract infection. But most of the severe coronaviral diseases that are prevailing in the human being are originated from the beta coronavirus group, and those are the SARS, MERS, and the present day SARS-CoV-2. And this SARS-CoV-2 actually causes the COVID-19 disease, and this SARS-CoV-2 is originated or evolved from the SARS-CoV-1 that was previously happened. And in this slide, okay. So now, how the coronavirus works, as it's already discussed that the coronaviruses are the shape of the coronavirus are spherical or elliptical in shape, and sometimes they are pleomorphic or doesn't have any specific shape in some cases. But in case of the SARS-CoV-2, it's spherical in shape and it has a helical symmetry, and it possesses outside the nucleocapsid, it processes a typical envelope-like structure, and this envelope-like structure produces crown-like appearance due to the presence of the spike protein, and that's why the name implied that the coronavirus, because the Latin name coronum means the crown. And the, unlike the other coronaviruses, these coronaviruses are also sensitive to UV light, and in high temperature, like greater than 60 degrees Celsius, an incubation period is 15 minutes is enough to kill the coronavirus, and it can survive the chill of 0 degrees Celsius up to 0 degrees Celsius. And the lipid solvents can effectively degrade the viruses, and that is greater than 75% alcohol-based hand sanitizers are effective, and other chlorine-containing solution like sodium hypochlorite, peroxyacetic acid, chloroform, etc., can be utilized as an effective disinfectant against the COVID-19 disease or coronavirus. And recently, it has been shown that the chlorhexidine, that is the principal component of most of the mouthwashes we use, they can also limit the viral, somehow limit the viral load in our mouth. So the using the US CDC or Center for Drug and Center for Drug and Diseases have released one bulletin that said that the chlorhexidine can be used to reduce the infection that are coming through the mouth into our body. Next slide, please. So, so now how this viruses enter the host cell to take control over them. Uh, Next, you can just click on yes. First, the virus come in contact with the receptor present on the host. Next, and and then it releases its genetic material inside the host body. Next, then it utilizes the host translational machinery. Note it utilizes the host translational machinery to produce the replication enzymes for its own, and then it produces next slide. Next, please. Then it produces multiple copies of its genome, which are packed into the new viral particles. Next, next. And during doing this, the host immune system causes the constant selection pressure along with the external stimuli like drugs, etc., that causes the mutation, and it changes some of the viral particles. And this mutation can lead to more virulent or less virulent strains of that particular virus species. These are popularly known as the different types of serotypes. Next slide. And after that it breaks down or disintegrates the whole host cell body and produces a huge load of necrotrophic tissue, and that are the principal material for other secondary infection. In this case, the secondary infection of the pneumonia infection are taking place in the lungs after the coronaviruses. And now the question is, what are the receptors in the host body that take part in this host pathogen interaction? Next slide. So these are the popular receptors present in the host cell uh, against the different coronavirus uh, species. And SARS-CoV-2 recognizes the ACE2, or which is called angiotensin converting enzyme 2, that's present in the host cell surface for their successful interaction with the host cell. Next slide. This is the structure of the coronavirus, and you can see there are four major structural proteins. These are the spike protein, nucleocapsid, membrane protein, and the envelopes. And among all these four proteins, the spike protein is most important because this spike protein actually take part in this host pathogen interaction. The spike protein again contains the two subunits H1 and H2, and these proteins are involving in the recognition reaction with the ACE2, or angiotensin converting enzyme 2. I'll come in detail regarding this specific reaction. Now, the genome organization, the structure, as we all know that the coronavirus is a positive stranded RNA viruses, and it has a unusually large RNA genome. Usually the RNA viruses don't possess that much huge structure of its genome as compared to the other RNA viruses, but coronavirus possesses the huge RNA structure as its genome, and it possesses two common proteins, that is REP1 and REP1b, which are responsible for the replication of the viral proteins. But the rest of the part is the structural proteins that produces the spike, membrane, envelope, and nucleocapsid, is popularly known as SMEN, for the production and organization of the viral particles. And if you see that SARS-CoV have relatively large structural protein organization, and that is the reason uh, it can produce so much variations, so much serological variations in their structural region, and it's a huge challenge for developing vaccine against the SARS-CoV-2, because it can change its structural region, because the size of the structural proteins and the structural regions is quite big. Next slide. Next slide, please. And This slide, the previous one, please. This slide tells that how these structural proteins are present in the coronavirus and how they are important. You can see the membrane protein contains around 2,000 copies, and whereas the nucleoprotein have the 1,000 copies and the envelope protein have the 20 copies. And if you see the spike protein, it has 100 copy per variant, and it has 10 nanometer in length, and it composed of 300 monomers. Just imagine just 10 nanometer size spike is composed of 300 nanometers. So how different combinations are possible in the spike protein? And this spike protein is principally important because this protein actually helps in interaction with the host cell, and it determines the immunogenicity of the host as well. And when the virus particle comes in contact, it takes only 10 minutes to get inside. It has an eclipse time of 10 hours, so we cannot identify the entry of the virus until like 10 hours, and it has 10 to the power 3 burst size. So after a single set of replication, it can produce 10 to the power 3 number of variants. Next slide. So this slide actually tells the internalization of the SARS-CoV-2, and it's very important and interesting as well. So as I told already, that ACE2 interacts with the S1 protein, the spike protein, and the spike protein has two domains. One is S1 and another is the S2. First, S1 interacts with the ACE2 for the recognition reaction. After the successful recognition with the virus with the host cell, there is a host serine proteases which is called TMPRSS, is a transmembrane protease S, and this transmembrane protease serine, and it cleaves the spike protein in the S1 S2 junction. So the S1 protein relieved, and it opens up the S2 it opens up the S2 protein, and S2 protein is nothing but a fusion peptide that fuses itself with the host membrane, and that allows the virus to release its genetic material inside the host body, and that is enough for the infection. And now, uh, if we understand the genetic structures, can you go to the previous slide, please? Yeah. If we compare the interaction between SARS-CoV-2 with the SARS-CoV, we can have an idea how the SARS-CoV-2 originated. So SARS-CoV has a single arginine residue where the TMPRSS or the transmembrane protease can cut the spike protein. But some modifications or some mutations taken place in this junction, so that in SARS-CoV-2 there is a three possible recognition site for the protease. One is arginine 685, arginine 683, and arginine 682. So it allows the host TMPRSS to cut the spike protein in three different sites. So it has a more flexibility than that of the SARS-CoV, and that is the reason why this COVID-19 disease is spreading so quickly and so vibrantly among the population, because evolution has taken place and mutation has taken place in such a way it gives the TMPRSS a better window for the cleaving site. Next slide, please. And this is very interesting that the genomic analysis of different SARS-CoV-2 from the different sources, like from the pangolins, from the bats, and from the human SARS-CoV-2, it clearly demonstrate how this evolution taken place. In the receptor binding domain of the spike protein, there are 6 nucleotide change. Next slide, please. Next slide, please. Can I go to the next slide? No, the previous one is previous one, please. Yes, this one. So there is a mutation in the RBD leading to 6 amino acid changes that high affinity binding of the ACE2, and these 6 amino acid changes actually gives the TMPRSS or the host proteases that window or that flexibility. And second point is the polybasic furin cleavage site. Just incorporation of a single proline residue in this region gives the better cleaving probability of the spike protein. And that is another reason why this COVID-19 disease is spreading so furiously. And the third most important modification that takes place in SARS-CoV to generate the SARS-CoV-2 is the addition of a O-linked glycan in the residue serine 673, threonine 678, and serine 686 that produces a mucin-like coverage. So this is the coronavirus, the envelope, and it the O-linked glycans produces is mucin-like covering sheet around it. It actually covers up the epitope for the epitopes that are utilized by our immune system to generate the immunogenic effect. So it seals the epitope. It seals its epitope, and this phenomenon or this technique is also adopted by the HIV virus. So the people, that's why there is a difference between the coronavirus and the SARS-CoV-2. SARS-CoV-2 help the coronavirus to evade the host immune system. Next slide, please. Next slide. Okay. Now, the ACE2 or angiotensin converting enzyme 2 is mostly present in our uh, nasopharyngeal region and also the endothelial region of different blood vessels, lung, kidney, and heart. And that's why the kidney, lung, liver, and like lungs, heart, brain, etc. These are the principal organs that are affected by the coronavirus. And now the question is, what is the actual function of this angiotensin converting enzyme in normally in the plant, normally in the human body? Actually, angiotensin is a peptide hormone that controls the vasoconstriction, or it controls the blood pressure as well as the renal secretion. It helps the sodium ion to retain inside our blood vessels. So firstly, the angiotensin 1 is a nascent peptide. It's converted by the ACE or angiotensin converting enzyme into angiotensin 2. And this truncated angiotensin 2 is sensed by the ACE2 and angiotensin 2 type 1 receptors, which are mostly involved in the lung health and as well as the blood pressure. So that's the reason why high blood pressure groups and the groups with the diabetes and the cardiological disorder have the higher chance or higher probability of getting this disease. At the same time, I want to note one thing: the smokers have the greater chance to be infected because ACE2 is highly expressed or overexpressed due to the smoking. So as there are so many, the ACE2 number increases, then there will be a greater chance of infection. So quit smoking as soon as possible. Next slide. So most of us, all of us know now that what are the different symptoms of the coronavirus. That is nothing new. But what is new is that in most of the cases, the coronavirus disease is said to be mild in 80% cases. They have the common similarity with the common cold and something like that. And after day 4 to 9, only 14% patients got the higher level of infection. They need the respiratory assistance, and only 5% cases they need the ventilation or severe cases. Now, and day 8 to 15, when the infection goes to the bloodstream, and it causes the multiple organ failure. And now the question arises, why in some population the infection is milder than other? Yes, immunity is definitely important part, taking an important part in this, because the people with the good immunity have the better capability of restrict the virus. But recently, one study has published in New England Journal of Medicine, where they have studied the genome-wide association study with the severely affected COVID-19 patients. And they found a very interesting finding that the single nucleotide polymorphism is associated with the high risk factor of the high risk individuals. And what are those? There are two locus in two different chromosomes in human chromosome. One is chromosome number 3 and another in chromosome number 9. In chromosome number 3, the locus is 3p21.31 is associated, they're associated six single strand gene, SLC6 at 20, AZTFL1, CCR9, FICO1, CXCR6, and XCR1. Most of them are different transporters and chemokine receptors or cytokine receptors, and they have found that the association of these six genes is highly related with the high risk individual. That the presence of this single strand gene in the chromosome locus 3p21.31 increases the risk of higher degree of infection or higher degree of symptom severity in the population. And second one is far more interesting, that is the chromosome number 9 and the chromosome locus 9q34.2, which is the locus for our ABO blood group, is highly related. They said this highly related with the risk of the coronavirus. And what is that? The blood group, the people with the blood group A have the higher probability of getting the infection than the other blood groups. And interestingly, the blood group O have the self-restrict or self-protective effect against the coronavirus. So people with the blood group A are somehow more susceptible, and people with the blood group O is somehow less susceptible to the coronavirus disease. But please don't think that the people with the blood group O are safe, absolutely safe from the coronavirus disease, because it's just a probability. Next slide. Okay, this is the infograph made by me in during March, and it tells the how the coronavirus disease are detected in different kits. And there are different methods available now, but the major methods are still the ELISA-based kit, the CRISPR-based, and the RT-PCR-based kits. The ELISA-based kit utilizes the blood sample and the antibody against this coronavirus spike protein. The engineered spike protein is used in the kit, and it determines or it detects the antibody inside the blood sample of the patients. And it has not been still approved by the FDA. And the CRISPR-based technique is a very interesting technique, as a very fast technique, where the Cas12 is guided such a way that the target, if the sample has the target DNA or the sample has the coronaviral RNA, it clips that RNA, and a sensitive molecule changes its color, and it's very fast. By changing the color of the sample, we can detect that the patient is positive or negative. But despite of all these techniques, the reverse transcriptase or real-time based technique is always very much sensitive. But the only drawback of this is it takes some time. The swab test is always the confirmatory test till now, but it takes some time, because after swabbing, the people has to isolate the RNA from the swab, and then it's converted into the RNA is converted into the DNA or cDNA by the reverse transcriptase enzyme, and then specific primers are used to perform the real-time experiment. So Why this technique is so sensitive? Because real-time PCR can detect a very small amount of the genetic material, because its sensitivity is too high. So the people, even if they are not originating any symptoms, can be identified by this real-time based technique. But other techniques, sorry, the other techniques is very difficult to identify the groups with a very low level of infection. Next slide, please. So this slide tells the spread of the disease, that we all know how the contact spread is taken place from people to people, and ultimately it produces the community spread, that we cannot identify the source from where the uh, spreading of the from where the spread of the disease have started. And unfortunately we are presently in a partial community spreading stage, because in the last year there are more than 17,000 cases are reported in one day. So you can imagine that the community transfer uh, may be partial, but it has already started. So next slide, please. Next slide, please. So, and this infograph was also made by me during the imposition of the second lockdown by the government, and to entail the importance of the lockdown. You can see the happy faces, and these happy faces, the people with the successful lockdown. But unfortunately, we cannot be so happy after so many of days, because maybe some of us are not following the lockdown properly, and we are in a stage of exponential phase. Now, very interesting slide. This is the sneezing dynamics. Erin Bromage, a leading scientist, has calculated the infection dynamics, that infection is proportional to the viral particles into time. So normal breathing can release 50 to 50,000 droplets per exhalation. Can you imagine 50 to 50,000 droplets? So you can realize it. If you wear a mask and a spec, you can find that a huge amount of vapor is accumulating in your glasses. So that the number of the droplets you are developing in a single exhalation. And from the previous viral disease, it is obvious, it is reported that at 20 virus particles are released per minute. And as I already told, the 1,000 particles are required to get infected. And during the speaking, it increases around 10 times. So 200 viral particles are released in a single minute. So if you are talking with a corona positive patient, so it takes only 5 minutes to get infected. And in the left side, it showed that how that huge amount of the millions of droplets are originating during sneezing. Next slide, please. Next slide, please. Next slide. Okay, so from the previous slide, you can understand why the wearing mask properly is important. At the same time, hand hygiene and maintaining the social distancing is the only way to protect us from the infection. And according to the European standardization, DIN EN 149, there are three different types of masks available. The FFP1, FFP2, and FFP3. FFP1 is can capable 80% of the particles to wipe out, and FFP2 can occupational exposure limit of 94% to 95% is popularly known as the N95 mask. And FFP3 is used to protect the toxic solid and liquid aerosols. It has the highest possible protection under EN 149, which has a limit of 99.5% effectivity. And this FFP2 and FFP3 are important to use for the healthcare officials or healthcare workers, and for the normal common people like us, is enough to wear a cotton mask. Next slide, please. So a three-layer cotton mask is best option for common people. So next, next. So one report from the University of Nebraska Omaha has tells that the cotton different materials that can be used as a homemade mask material and the effectivity of this material. But the wearing mask properly is very much important. Next slide, please. You can click on continuously. So these are not the method by which the people are wearing mask. So the correct way of wearing mask and spec is next slide. Next, please. The correct way to wearing mask is you have to wear your specs over your mask, not under your mask. Next slide, please. Next slide. And now I'm going to previous slide. Okay. Now I'm going to tell some of the major drugs administered against COVID-19. So it is some of the drugs have already discussed by my previous speaker, and the detailed about the vaccine and vaccination procedure have been also discussed by my previous speaker. But I want to just list out 9 topmost drugs that are used in last couple of months. That is remdesivir. This is the RNA-dependent RNA polymerase inhibitor. It is the most successful drug that ever been used against the COVID-19. And everybody, we all know the chloroquine and hydroxychloroquine. It was first reported by a French group by a very small sample size study, but later on it has discovered that this chloroquine and hydroxychloroquine is not that much effective against the COVID, but it helps somehow because the glycosylation, it imposes the glycosylation of ACE2 receptor, and also the alkalization of the cell cytosol retards the viral replication. At the same time, it can help in better antigen presentation also. And another drug is chemostat mesylate, is a serine protease inhibitor, and tocilizumab is a humanized antibody, is widely used in China, and it controls the cytokine storm. The cytokine are the very important chemokine that are released in our bloodstream due to immunogenic effect, but uh, ex, the ex, expression of too much cytokine in a short period of time produces the cytokine storm, which is also called hypercytokinemia, can causes the severe effects like multi-organ failures and something like that. And in severe cases of the COVID patients, we observed that the cytokine storm were there. And to control the cytokine storm, tocilizumab is an effective drug. Lopinavir and ritonavir, this combinatory drug are generally these are the protease inhibitor, and they are used against the HIV virus. And they are also, as there are similarity between the HIV and the SARS-CoV-2, they are also administered against the SARS-CoV-2, but it was found that they are not that much effective unless it's utilized along with the interferon beta. Nitazoxanide is now tested. No report is still now in our hand. And another drug is dexamethasone. This drug is under trial of recovery program by UK. Recovery is the randomized evaluation of COVID-19 therapy, and it was found that this dexamethasone in a very low dose, that's like 6 mg dose, it can reduce the death rate for the terminally ill patient, like 1 to 1/3 in case of the ventilator, patients with the ventilator. And it also retarded the death rate of the patients who needs the respiratory assistance up to 1/5 times. But it has no effect. It has no far effect so far effect on the people uh, who do not have such severe symptoms. So it's very clear that probably the dexamethasone is not an anti-COVID drug, but it somehow uh, helps the patient with the respiratory assistance not being like the situation, to control the situation from the more severity. And ninth one and most interesting one is the convalescent plasma. The neutralizing antibody are extracting from the COVID-19 survivors, and they are utilized uh, in case of they're injected in different terminally ill COVID patients who fail to activate their own uh, immunogenic immunogenicity or immune activity. Next slide, please. So, from the discussion is very obvious that we all understand that COVID 19, we don't have any effective drug or effective vaccine against the COVID 19, and the generation of the vaccine definitely takes time. This is not a one night job. It takes a time, and it has some procedures to develop. And also the people all around the world are trying their best to develop the effective drug or contain the disease, but there are a huge responsibility of us as a responsible citizen to protect ourselves and our loved one from this devastating disease. So please follow the all the guidelines produced or given by the WHO and ICMR that we all know, because everybody uh, recapitulate it in every time, but we are somehow misfollowing it, and are not following the all the guidelines properly. So it's completely our responsibility to follow and habituate in this new normal to contain the disease. Will definitely survive, and all the humankind will definitely survive and fight against this biggest challenge in the recent past. Next slide, please. So thank you for your patience, and definitely wearing mask. Thank you.
Thank you very much, Dr. Anirban. Thank you very much for your lucid and very descriptive talk. I think all the students, especially, have very much benefited from your talk. And as for the question answer session, I am sorry to say that we have received a lot number of questions, but due to very much short of time, I will be able to put very importantly key questions to the two speakers. My first question is to Dr. Anirban Kaur. What do you think is the chance of a second recovery from a second infection from a already recovered patient?
Okay, it's very interesting question, and there are every chance of the re-emergence of the disease, because the virus is SARS-CoV-2, but there are several serological varieties of these viruses. Like when it originated firstly in Wuhan, China, there is a mother strain that is called the zero or O, and after that there are several different variants like A2A, A2, A23, and B1, B2, like different serological varieties are there, and I already told that the mutation is going on continuously. It's not has not stopped. So there is every chance of getting the new variant of the same virus. So the people, for example, the people are recovering from a particular type of serovariants can readily infected by other serovariants after their recovery. And another point I want to mention um, in this direction is that as I told that there are four different groups: alpha, beta, gamma, and delta. So most of the coronavirus so far reported infecting human races are from the alpha and the beta origin. But there is a readily evolving group, that is the delta group, are growing so fast. So there is every chance of getting a new coronavirus in the recent future. Also, this is another context.
Okay. Thank you very much, Dr. War. My next question is to Dr. Gaurav Sarkar. Do you really think it is very much possible to design a vaccine which will have a broad spectrum effect on a wide range of people, as we know that there are many type of genetic get-up among different carriers or patients. So is it possible to design a broad spectrum vaccine?
Yes, because if we can include epitopes as many as possible from various strains, that's what I was talking about, the broadly neutralizing antibody. So it's yes, it's possible to make a vaccine which can be effective not only against SARS-CoV-2 but the entire the coronavirus group, all the members of the coronavirus group. It depends on how I mean the vaccine, how many epitopes are being represented in the vaccine, how many B cell epitopes and the T cell epitopes are being represented by the vaccines. As many epitopes are there, the polyclonality of the, of the protective immune response will be broad, will be wider, and that will, of course, that will give protection, wider range of protection. Yeah,
Yes.
that's it.
Yes, I guess that this is a question which is still to be answered because the vaccine research is still going on. And I think we really don't know the answers. Yeah. Okay, so I thank all the speakers, and although we had many questions, but due to very short of time and we had technical issues right from the beginning, I'm sorry that I had to shorten this portion. So now it is the final. I would like to give the vote of thanks as we have already come to the end of the ceremony. So well, now finally, as we have reached the completion of our two-day national webinar, I, on behalf of the entire Department of Botany, take the opportunity to thank all our notable speakers of yesterday, Swami Kamalasthananda Maharaj Ji, Dr. Priyanka Bhomi, and of today, Dr. Anirban Bhor and Dr. Gaurav Sarkar, for their enlightening talks, which have definitely augmented our knowledge about COVID-19 and also helped in increasing our mental spirits to win the war against COVID-19. Well, once again, I would also like to extend my sincere thanks to our principal sir, Professor Prashanto Bhattacharya, for his constant encouragement in conducting this webinar. We are also grateful to our IQAC coordinator, sir, Professor Tanmoy Mandopadhyay, for his invaluable advice and his constant support throughout the process. I would like to thank our librarian, Dr. Pronovi Porel, for her endless support and cooperation. We are really indebted to you, Dr. Porel. I would like to thank our librarian. I would like to again thank our IT committee convener, Dr. Robiul Alam, and convener of academic subcommittee, Dr. Shucheta Joy, for their support. I would also thank my joint conveners, Professor Jaita Shaha and Deputy Convener Professor Shagoto Mukherjee, for constantly putting their effort in making their webinar a great success. I would also thank my departmental faculties, Dr. Shelly Sinha, Professor Arjo Dash, our laboratory instructor, Mr. Sharbashish Banerjee, our laboratory assistants, Mr. Devobroto Pandit and Mr. Shubrata Ado, for providing their mental support from behind. I thank all the participants for their overwhelming response and participation in making this event meaningful, though we had met serious technical problems. So looking forward to win the war against the submicroscopic enemy COVID and resuming our normal life in an unlocked world very soon. Thank you all. Stay safe and stay well.